Hello everyone. Today, firstly, we have something called BLAST. What is BLAST? BLAST is a basic local alignment search tool. It basically finds regions of local similarity between sequences. This program compares nucleotide or protein to sequence database and also it calculates the statistical significance and matches. BLAST also can be used to infer functional and evolutionary relationships between sequences, as well as help to identify members of gene families. Next, we're gonna go to the home page and we're gonna press on right here, Protein BLAST. After that, something like this is gonna appear. So we're going to use that box over there to copy our protein sequence that we just talked about, the 3BEQA. Now we're going to apply this for 3BEQA and 3BEQB. Now because they're both identical, I'm going to show you only the first one, the 3BEQA. In the setting, we need to change the database to non-redundant protein sequence, NR, and also we need to put PSI BLAST. Next, we press on BLAST. Alright, so that's what's going to appear. We have the conservative domain and also the query sequence. We have these lines. Now, we have mostly the red lines. The red color indicates the most similarity to our reference, so it's the best one. So now we press on any of these lines and something like that is going to appear. So this is basically the informations, the sequence ID, the length, the number of matches, and also we have the percentage. As we can see, the identity here is 100% similar. The positiveness is also 100%, and there are 0% gaps, which is a very good thing. It's the IntraProScan. IntraProScan is a software package that allows sequence such as nucleic acid or protein to be scanned against IntraPro signatures. What are signatures? Signatures are basically predictive models provided by several different databases that make up IntraPro consortium. So the IntraProScan is a tool that combines different protein signatures, recognitions, methods, and it all makes it into one resource, and that is what we will do right now. We go to IntraPro, we type it in Google or wherever search tool that we want. We will also have a box here, so we're going to use that box to paste in our sequence, our protein sequence, the 3BEQA as well, and we're going to search it. So something like that is going to appear. As we can see, we have the glycoside hydrolase and also the silidase. These two are basically similar to neuraminidase, and we can see the information about them if we click on that little box on the left-hand side. If we click on it, we're going to have this, the whole description of it, the family relationship, everything about it. Next, we're going to talk about CSA, or Catalytic Site Atlas. What is Catalytic Site Atlas? It basically provides catalytic residue annotation for enzymes in the protein data bank. This website is very trusted and it will be updated every month, so all the information that we have, all the protein data bank, all the enzymes will be there. So how to do this? Our 3BEQ without any domain, and we're going to search it. What we will find here is the name, the species, everything about the 3BEQ. And also, the two chains that it has, the chain A and chain B, is going to appear. As we can see here, they're both identical. Next, we will move to the last part of our presentation, which is the ALN and DND. What is ALN and DND? Is basically a multiple source alignment that gives various kind of results after alignment. ALN files contain the next alignment, and DND contains distance information. How to do it? We have one software that can do it, which is called the Cluster X. So we go to File, we open we load our sequence. We have multiple sequence here. We use multiple sequence for better graphical view, including our 3BEQA and 3BEQB. So we just load our sequences. As we can see here, we have the sequences. It's not aligned, so we go to alignment, we do complete alignment. Press OK, and we will see something like this. Alright, so as we can see, the color-coded alignment gives a better graphical picture as the amino acid or protein residues are assigned color based on the physical and chemical properties. Next, we're gonna use a different tool which is called Mega. And we will open our file, which contains the DND that we got from Cluster X. That's our DND file. We load it in, and then we will have something that looks like a tree or a chain. We're gonna press on this one right here, alright, and we see all the sequences that we have, including our two main sequences that we need, the 3BEQA and 3BEQB. So now I'm going to show you the ALN, how it appears. It's just like this. And that's all from us. Thank you very much and enjoy.